Hello, in this video we're going to look at the Nobel Prize in Physiology or Medicine from the year 1906 awarded to Camillo Golgi and Santiago Ramón y Cajal in recognition of their work on the structure of the nervous system. First, some about Golgi. From the official Nobel Prize site, during the 19th century, researchers learned that to stain tissues without study under a microscope. In the 1870s, Camillo Golgi discovered that the nerve cells could be stained with silver nitrate. This led to the groundbreaking studies of how the nervous system is structured and functions. Golgi maintained that all nerve cells in the nervous system constituted a continuous interconnected network. This led to a protracted disagreement with Santiago Ramón y Cajal, the leading proponent of another opinion, and eventually Cajal's ideas prevailed. This is coming from the facts section from the Nobel Prize site on this Nobel Prize. There's more information. There's a speed read also on this subject, and there are 3 articles, one on Golgi, one on Cajal, and one on them both. And the links you can find here. There will also be links in the description of the video. But to the Nobel Prize site, the 1906 Prize for Medicine or Physiology, and there you can find the rest. Some biographical information summarized by me. Golgi himself stated that Bizzozero greatly influenced him and his methods of scientific research. At that time, most of his investigations were concerned with the nervous system, insanity, neurology, and the lymphates of the brain. Already while working at the hospital of San Matteo, Golgi became interested in the investigations of the causes of malaria, and he must be credited for having determined the 3 forms of the parasite and the 3 types of fever. After prolonged studies, he found a way of photographing the most characteristic phases in 1890. Golgi was a famous teacher. His laboratory was open to anyone anxious to do research. He never actually practiced medicine, but directed the Department of General Pathology at San Matteo Hospital, where young doctors were trained. He also founded and directed the Istituto Scienziatico Facinogeno of the province of Pavia. Golgi was rector of the Pavia University for a long time and was also made the senator of the Kingdom of Italy. He was an old man during the First War, but assumed the responsibility for a military hospital in Pavia, where he created a neurological, neuropathological, and mechanico-therapeutical center for the study and treatment of peripheral nervous lesions and for the rehabilitation of the wounded. However, the work of the greatest importance which Golgi carried out was a revolutionary method of staining individual nerve and cell structures, which is referred to as the black reaction. This method uses a weak solution of silver nitrate, particularly valuable in tracing the processes of and the most delicate ramifications of the cell. All through his life, however, he continued to work on these lines, modifying and improving his technique. Again, coming from the official site, biographical section, and here you see the article about Golgi himself, Life and Discoveries of Golgi. There's also one on Cajal. Here you see his Nobel Prize diploma. And here you see, and this is coming from one of the articles, the beautiful pictures they made. This is a Purkinje cell. This is cerebral cortex. This is also a Purkinje cell. And this also part of the cerebral cortex. Most of them, of these illustrations, were made by Golgi and his collaborators. You see the hippocampus. You see a stained part of the dendrite branches. Again, the hippocampus. This is all stained by the Golgi method. You see him in his office, his laboratory. Here again, beautiful drawings. You can read what it is. Cerebral cortex is this one. Typical rosette shape of a malarian parasite. There's this one. Sorry. A tendon organ, that's this one, and here that is on the right, completely the internal reticular apparatus. Beautiful pictures. And you see where he worked. Yeah, okay. Then on Santiago Ramón y Cajal works. Well, again, coming from the official side, as always. Our bodies are controlled by the interaction between the brain and the nervous system, which extends throughout the body. Camillo Golgi's discovery in the 1870s that nerve cells could be colored using silver nitrate opened up new opportunities for study. Ramón y Cajal began using this method in 1887 and achieved many groundbreaking results in the years that followed. This includes proving that each nerve cell is an independent entity and nerve synapses transfer nerve impulses from one cell to another. You see the link to the work section, and here to retweet, and here to the article on him and Golgi. There's some biological information also coming from the site, Nobel Prize site. In 1880, he began publish scientific works, of which the following are the most important. My Spanish is not so well, so I just read the English translation. Manual of Natural Histology and Microscopic Technique, 1889. Summary of this manual recast with addition appeared under the titles Elementos de Histologica, Elements of Histology, a manual of general pathological anatomy, 1890, and in addition may be cited Les Nouvelles Idées sur la fine anatomie des centres nerveux, New Ideas on the Fine Anatomy of the Nervous Center, the textbook on the nervous system of man and the vertebrates from 1897 to 1899, the retina of vertebrates from 1894. Apart from these works, Cajal has published more than 100 articles, especially on the fine structure of the nervous system and especially of brain and spinal cord, including that of muscles and other tissues, various subjects in the field of general pathology. These articles are dispersed in numerous Spanish journals and in various specialized journals of other countries, special French ones. Some articles in Spanish by Cajal and his pupils appear in the Revista Trimestral de Histologia Normal y Patológica, quarterly review of the normal and pathological history from 1888 onwards. A continuation of them appeared under the title Communications of the Laboratory for Biological Research from the Madrid University. Cajal studies on the structure of the cortex of the brain has been partly grouped together and translated into German by Bresser, 1901. This is the link to the information. This is summarized, of course. If you follow the link, you can get the complete information and this link to follow to the article about him. Also, Cajal made very beautiful drawings and pictures to illustrate his books. Here you see various types of nerve circuits and how they are layered. Pyramidal cells of the cerebral cortex and of part of the spinal cord. You see this coming from his articles or his books. Here, these are from one of the articles on the Nobel Prize site. If you want to see them incomplete, follow the link. Neurological cells from a cat. Neural connections on the right side. Cerebral cortex. And another picture of the cerebral cortex. Optical nerves in the front, human frontal cortex. Well, then the award ceremony speech by Professor the Count K.A.H. Müller, summarized by me, of course. But a far greater complexity appears if the impulse continues to be transmitted and reaches the centers of the consciousness. The impulse progresses along nerve tracks which follow complex pathways until it reaches the surface of the brain, which is the cerebral cortex. For consciousness, in man at least, is exclusively located in this area. Until it reaches this area, the transmission of the impulse must remain isolated, otherwise If other pathways corresponding to other parts of the skin become involved, the site of injury may be incorrectly located. If a painful sensation is eventually perceived, limited to the irritated area of the skin, this sensation may in turn give rise to a number of different activities within the central nervous system. It can give rise to thought and action. In this case, painful sensation can be linked with memory traces from earlier experiences obtained in various ways and stored in various areas of the brain. This process presupposes a system of connection between different parts of the cerebrum. Finally, stimulation may occur of other cells in the cerebral cortex which control voluntary and conscious muscular activities. When these occur, these cells produce impulses which provoke muscular reactions appropriate to the circumstances. The mechanism of transmission, which we have briefly outlined, correlated with functional phenomena, will, I trust, demonstrate the complexity of the mechanism required for the functioning of the nervous system. Our present knowledge of this mechanism has been acquired in a number of different ways, by research in the field of comparative anatomy, by studying development of the nervous system, by physiological experimentation, etc. The way which would appear to be leading most directly to direct knowledge is direct anatomical observation remained impractical for many years. It had been shown that the nervous system contained, apart from blood vessels, etc., supporting substance composed of cells and fibrous structures, and the element proper also composed of filaments and cells, which at different places showed a different appearance. The nerve cells, which for good reasons were considered as stages and foci of the nervous pathways, were found to be concentrated in those areas of the central nervous system which are characterized by gray pigmentation. It was often difficult, however, to distinguish between the real nerve cells and the cells which made up the supporting substance. It was also known that many nerve cells give off cellular processes in varying number, among which one in particular, by reason of its special appearance, was to believe to give rise to true nerve fiber. Unfortunately, it was not possible to follow this process for a very long distance along its pathway. As for the other cellular processes, which ramified very quickly, they were the object of guesses rather than direct observation. Our knowledge of the nerve fibers was also, to a great extent, incomplete. In the wide areas of the central nervous system, grouped nerve fibers were seen, similar in appearance to the peripheral nerve fibers. But to what extent did those of the first group prolong themselves into those of the second group or link up different centers in the central nervous system? Did these fibers produce ramifications or not? Did they communicate or not with other fibers? Such were the questions which required answers. It should be remembered in particular that almost nothing was known for certain of the relationship between nerve fibers and nerve cells. The central nervous system appeared as a confused mass of filament, each as fine as the thread of a spider's web, and of microscopic cells armed with cellular processes. Golgi's silver impregnation proved crucial and was taken up by Cajal with much success. And this is coming from the official Nobel site. Here you see the link. This is summarized, of course. If you want to read the complete award ceremony speech, follow the link. Well, that was it for today. Next time we will continue, as is next week, with the 2 lectures given by Golgi and by Cajal when they received the prize. Thank you for watching, and maybe till another time.